Good morning, good afternoon, and good evening, everyone, and welcome to our webinar titled Multimodal Behavioral Assessment After Experimental Brain Trauma. This webinar has been sponsored by Colborne Instruments, PanLab, and Harvard Bioscience, so a big thank you to them for helping to make this event possible. Joining us today, we're very fortunate to have Dr. Corina Bondi, an assistant professor at the University of Pittsburgh. Her research focuses on experimental traumatic brain injury, the resulting cognitive deficits, and potential avenues for developing therapeutic approaches to treatment. So, without any further delay, I am very excited to welcome Dr. Corina Bondi. Corina, thank you so much for being here with us today, and the floor is yours whenever you're ready.
Great. Well, thank you so very much for the opportunity to present some of our work today, carried on at the Stafford Center for Resuscitation Research at the University of Pittsburgh. And thank you for everyone who has been tuning in from literally all over the world. I've seen not just stateside, but also Europe and South Africa and Asia and so forth. So that's wonderful. Much appreciated. I have no relevant financial or non-financial interest to disclose. And as Sarah mentioned in the introduction, our lab focuses on executive and cognitive dysfunction after traumatic brain injury. So as a brief introduction, for those of you who might not be familiar with the field of TBI, there are basically over 2.2 million emergency departmental visits in the United States from TBI, and there are over 10 million TBIs worldwide every year. In the US, it leads to over 280,000 hospitalizations and over 50,000 deaths, as well as direct and indirect medical costs of over $76 billion. The symptoms after TBI come in different categories, but one of the main categories they were interested in are cognitive symptoms. So behavior or mood changes, alterations in memory, concentration, and attention. Of course, there are also more severe symptoms such as persistent headache, seizure, loss of coordination, confusion, and agitation. Everybody is at a risk for TBI, really. Of course, it can be an accident, can happen anytime, but some of the highest risk groups are children under the age of 14 and the elderly over 65, because of course they have less balance and stability and they're more likely to fall and hit their head. So when we think of cognitive function in traumatic brain injury, we think of different brain regions, but one of the main ones is the prefrontal cortex, or the PFC. And the PFC-dependent executive function refers to a collection of related yet distinct abilities to provide for intentional, goal-directed problem-solving action, therefore basically organizing and directing all top-down control of cognitive activity, emotional responses, and overt behavior. Executive dysfunction is a common outcome in TBI, and it's often seen as specific deficits in planning, cognitive flexibility, as well as behavioral inhibition and poor organization of learning, memory, and language formation. And what you see on the bottom are three imaging cases of patients with frontal lobe damage, ranging from milder on the left side to a more severe contusion on the right side, all of them afflicting the prefrontal cortex. So, considering the intricate connectivity that's either direct or indirect of the prefrontal cortex with various brain regions, it has been an interest in the TBI field to tease out the possible mechanisms through which the frontal cortex may affect cognitive function, especially given the fact that it is connected to different and many other brain regions, from the striatum to the ventral tegmental area, substantia nigra, and so forth. This is a slide that I like. It's called Et Pluribus Unum, or Out of the Many One. Basically, several kinds of knowledge and afferent innervations combining to produce a sophisticated behavior. For a long time, it has been thought that the rat frontal lobe may not be very advanced, and it doesn't really do too many things. But a lot of research in the past 20 or 30 years has shown that, especially when you look at the green areas, right, the agranular cortex, you have the human brain on the left, the macaque monkey in the middle, and the rat on the right. A lot of the brain regions are similar, and they basically intimately influence behaviors in similar ways, ranging from the function of the anterior cingulate cortex, infralimbic cortex, prelimbic cortex, and so forth. So in the clinic, executive function is often tested through a variety of tests that are basically the cornerstone of assessing dysfunction in patients with a variety of conditions affecting the frontal lobe. One of the tests is the Wisconsin card sorting test, and in which patients are asked to find the correct classification principle across stimulus dimension by trial and error and examiner feedback. And there's a dose-response effect of TBI severity on Wisconsin card sorting test performance. So basically, they have to match the target card based on form, color, as well as number. And once they reach criterion on a dimension, it switches to the next dimension. There's also the continuous performance test, in which it measures sustained attention, which is highly sensitive to brain damage or dysfunction. It is a core deficit in the number disorder, and it's based on the interaction of cortical, frontal, temporal, parietal, and subcortical structures. Also, TBI alters continuous performance test performance. So one of the tests that we use in the laboratory is a test of sustained attention called the 3-choice serial reaction time task. It takes place in operant chambers. It is analogous to continuous performance test in humans, which is a variety of the 5-choice serial reaction time test. The operant chambers consist of a food dispenser and a trough on one wall, and 3 identical cue nose poke holes on the opposite wall. Brief light cues are randomly presented in one of the cue ports, and the rats perform the task for a 30-minute session. It takes us 2 to 3 months to train the animals on this task, and they need to reach higher than 70% accuracy and lower than 20% omissions. We measure the accuracy as a percentage of correct responses, the incorrect responses, again, the omissions, and the number of prematures, which are the number of cue nose pokes during the intertrial intervals. So we wanted to test the effects of TBI delivered as controlled cortical impact on the 3-choice serial reaction time task. So one of the methods through which we deliver TBI in our center, although we literally employ the majority of the TBI models, but one of the ones in our laboratory is a controlled cortical impact injury. The device was developed by Dr. Edward Dixon at our center, and basically animals are anesthetized, affixed on a stereotaxing frame, and you can do either closed skull or open skull over the dura mater, and a pneumatic piston will impact the surface of the brain at a predetermined angle or duration or depth. The most standard type of injury that has been done typically in TBI has been parietal cortex injury. And so we started with that for our test in both normal cycling females, particularly because women, when they suffer from a TBI, are at different phases of their menstrual cycle, as well as the fact that in previous literature in the TBI field in animals, it was found that the estrus phase cycle for motor and spatial learning wasn't really affecting outcome. So we're using normal cycling females versus males. Again, it took 2 to 3.5 months for the animals to be trained from 15-second cue to 0.3 second cues. So they have to really orient to the cues. Then they undergo surgery, TBI, at 2.8 cortical millimeter deformation depth. And then they're given 2-week break for recovery, and they were retested on the 3-choice task at days 14 to 24 post-surgery. So the 2 left panels are females, and the 2 right panels are males. Accuracy on the top row and omissions on the bottom row. And this data are training data prior to surgery for the 6 stages of the training levels, again ranging from 15 seconds to 300 milliseconds. And our data look exactly how they were supposed to look based on the literature. Basically, the animals reach the 300 millisecond level with 75 or 80% or higher percentage of accuracy and less than 10% omissions for both females and males. So in terms of post-surgery performance, we have females on the left, black and purple, males on the right, in black and orange. And the time points at the beginning of each graph represent the baseline averages. So basically the last 6 baseline sessions for each rat, followed by 2 weeks later, post-surgery testing for 10 days. And we found that sham surgeries, basically animals who went through the surgical procedures without the actual impact, 2 weeks later retained their level of 70% or higher accuracy for both females and males in this task. However, both females and males were significantly impaired in terms of percentage accuracy in the 3-choice task, and it remained sustained throughout the 10 days of post-surgery testing, therefore suggesting reduced sustained attention and vigilance. We also wanted to see, considering that we give our injury on the right parietal cortex, whether there was a laterality in terms of how these rats perform. So as the rats sit in the operant chambers and they focus their attention to the left, center, or right cues, we broke down the accuracy based on performance in those 3 specific cues, left, center, or right. And we found that animals seemed to perform particularly worse when it came to the cue appearing randomly in the left side of the operant chamber, so opposite to where their injury was, which is interesting because it seems that it would be a rodent analog of visuospatial neglect. You can see in the top 2 panel that accuracy performance in the left cue goes from 70% accuracy or so down to 30% accuracy on the first days post-surgery testing. In females, as the 10 days go on, it seems to start recovering. However, in males, it does remain more sustainedly affected. So in the clinic, patients that have visuospatial neglect are given visual scanning therapy, where they're forced to keep training their gaze towards the side that is affected. So we were wondering if possibly, interestingly, our females may benefit from that, as they have to basically keep moving their head and keep looking to the left side as the 10 days of post-surgery testing go on. So operant behavior is obviously great. It offers an objective and computer moderated means of assessing animal behavior, but we're also interested in testing cognitive function in a more kind of natural foraging environment for the animals. So one of the tests that we do for executive function and behavioral flexibility in rats is called the digging attentional set shifting test, or the DASD. It is a 2-choice discrimination using complex stimuli that differ along 2 perceptual dimensions, odor or medium. It was developed by Verity Brown, currently at University of St. Andrews in the UK, a couple of decades ago. And half of the animals start with odor as a relevant cue, and the other half start with medium as a relevant cue. You can see on the bottom right panel an image with the different pots and a pile of the small pieces of Honey Nut Cheerios that they like to work for. So in the case of an animal starting with odor, really briefly, they have to go over a course of 2 to 4 hours through 7 increasingly difficult contingencies. And for example, at the beginning, they have to dig in clove and not jasmine. Then we introduce an irrelevant dimension, in this case, medium. So they have to keep digging in clove and not jasmine, regardless of whether the pots contain raffia or metallic confetti. For each stage, they have to get 6 consecutive trials, correct trials, to move on. This is followed by the first reversal, where jasmine becomes positive and clove becomes negative. Then an intradimensional set shift with its own reversal, which basically it's a whole set of new stimuli. And up until this point, the animals form and reinforce a cognitive set. So for the extra-dimensional set shift, medium becomes relevant and odor becomes irrelevant, and they have to dig in velvet, regardless of the odor on the pots. And this is followed by its own reversal. So Dr. Brown's lab has shown in the first pivotal papers that whenever they lesioned with ibotenic acid the medial prefrontal cortex, and they tested the animals on the AST, on the y-axis of the top right graph, you have trials to reach criterion, and on the x-axis, you have the 7 stages of the task. And MPFC lesion specifically impaired the extra-dimensional set shifting performance by raising the number of trials to reach criterion. They followed that up with a study in which they lesioned the orbitofrontal cortex, which has been known in humans to be involved in reversal learning. And they found on the bottom right, you can see on the graph, that specifically the 3 stages for reversal learning were impaired in this case, and not the extra-dimensional set shift. So this was sort of some of the first papers that started to suggest that even though these are rats and they have a rat prefrontal cortex, different subregions of the frontal lobe in rats intimately regulate facets of the executive function and behavioral flexibility in these tasks. Since then, in the past 20 years, the digging AST has been used in over 120, 130 studies, and outside of the TBI field, where different laboratories have looked at schizophrenia models, different other dopamine manipulations with antipsychotic drugs, cholinergic manipulations, noradrenergic manipulations, serotonin in models of depression, and of course, a variety of excitotoxic lesions, showing that different pharmaceutical compounds can either impair or improve performance as hypothesized. So in our studies, we were also interested in changing or moving around the location of the injury, particularly because traumatic brain injury is so heterogeneous and no two injuries in humans look alike. People can hit their head in different locations of their skull, and oftentimes similar symptoms can occur with different types of injuries. And if you think of someone who might hit their head on the windshield in a car accident or fall off a bike or off the steps, the frontal lobe oftentimes is clearly affected. So we wanted to look at digging AST performance and compare what happens whenever we injure the parietal cortex over the hippocampus or the frontal lobe directly. So generally speaking, we give the surgery at day 0. The animals are allowed to recover for 3 weeks, and then they get put on the mild food restriction to increase their propensity to work for those small pieces of Cheerios, and then they get tested at 4 weeks post-surgery, which generally, as best as possible, it equates to about 2 years post-injury in humans. So in the first study, we looked at parietal control cortical impact effects on the digging AST task. We had naive animals that didn't even undergo surgery, sham surgeries, as well as 3 levels of injury just to see how sensitive the task is to injury. We had 2.6, 2.8, and 3.0 millimeter cortical deformation depth. And we found that it was specifically the 2.8 and the 3.0, so red and blue, that impaired performance by raising the number of trials to reach criterion on the extra-dimensional set-shifting task, ED, and the third reversal. We also look at total errors, just like in the Wisconsin card sorting test, for example, and we found that the 2.8 cortical millimeter deformation depth group significantly increased errors on the first reversal compared to the naive group. We followed up with confirming the different levels of injury by looking at the cavity that was formed post-surgery. And you can see in the upper panels the different levels of cortical lesions ranging from 2.6 to 3.0. And the 3.0 cortical deformation depth rendered a significantly larger lesion volume than the 2.6 cortical deformation depth, with the 2.8 being in the middle, which again is to be expected based on the literature. So we had another study in which we wanted to start looking at different types of therapies that may reverse these types of deficits. So we published a study a little more than a year ago where we looked at augmenting acetylcholine with galantamine, which is basically an acetylcholinesterase inhibitor, as well as a modulator at the cholinergic receptors. And we found that it restores executive function after parietal TBI. So if I can direct your attention to the first reversal, TBI plus vehicle, which is the black bar, was significantly worse in terms of trial to criterion compared to sham. And the 2 different doses of galantamine that we gave, 1 or 2 milligram per kilogram, significantly normalized performance on the first reversal compared to the animals that were injured but only got vehicle. In a different study that's currently under review, we also looked at augmenting serotonin with citalopram, which is a selective serotonin reuptake inhibitor, and looking at the effects of either citalopram alone or housing animals in environmental enrichment, which is a known kind of preclinical animal of neurorehabilitation, in which animals are placed, instead of their standard cages, are placed in these larger cages with multiple levels and 10 to 12 animals at a time. So it basically has kind of the rehabilitation component. They have larger groups with different toys and chutes and ladders, so they get to interact with their environment, and it's found to be very beneficial both in the TBI field and outside of the TBI field. And we wanted to see if the combination therapy may augment reversal learning recovery after parietal TBI. So I know that this is a busy slide, but if we can look at extradimensional set shift stage and the third reversal stage, which are the last two stages, basically just like in the galantamine study, the TBI vehicle animals, which were housed in standard housing, which is the clear bar with hashed lines, they were significantly worse than sham animals by having larger trials to criterion. Then TBI animals, they receive either citalopram or environmental enrichment housing, rendered significant improvements compared to vehicle-treated TBI animals. Interestingly, for the third reversal learning, which is the very, very last set of bars, the combined therapy, so the solid black columns, the combined therapy augmented performance from either citalopram or environmental enrichment because it was significantly better than the individual treatments alone. So like I mentioned, we also wanted to see digging attentional set shift performance after frontal injury. And we did a similar study like we did with the parietal TBI. We had sham animals as well as 3 levels of injury, 2.0 ranging to 2.4 millimeter deformation depth. And you have to go a little bit more shallow because the tissue in the frontal lobe is obviously smaller, the area. So the 2.2 and the 2.4 millimeters deformation depth significantly worsened performance on the extradimensional set shift stage compared to both sham and the 2.0 millimeter deformation depth TBI group, suggesting that indeed the moderate to severe frontal lobe injury was detrimental for executive function in this test as well. So moving forward, we wanted to take a look at how frontal lobe injury affects performance in a multimodal assessment of attentional set shifting. To the best of our knowledge, this hasn't really been done. Typically, laboratories will use either operant set shifting or digging set shifting, but we wanted to see if the two modalities were sensitive towards TBI and to each other. So in the operant chambers, animals were trained for 4 daily sets of attentional set shifting. Basically, they have to focus just on the left and the right cues for a food reward. They got moderate frontal lobe injury. And the behavior itself, it takes approximately a month to train. They have to focus their attention on either light or side. So in order to move from one set to the next in a given day and basically undergo 3 executive set shifts, they have to get 10 consecutive correct responses. So it might be light, light, left, right, or left, right, light, light, and so forth. Again, after training, they underwent surgery, 2 weeks of recovery, and then they're retested between days 14 and 23, following which they are trained and testing on the digging set shifting task, the days 24, 27. At the same time, some of our cohorts were given throughout the month post-surgery chronic osmotic mini pump administration of milnacipran at 30 kilograms per day, which is a dual serotonin norepinephrine reuptake inhibitor. So anecdotally, basically another antidepressant. It is an FDA-approved drug for fibromyalgia. So there has been some interest in the clinical community to take a look at its potential effects to restore cognitive function. So we're looking here at trials to criterion on the operant set shifting test. Total trials to criterion on the y-axis, the pre-insult average of the last 5 sessions per each rat as the first time points, basically with no difference among groups. Then 2 weeks after recovery, the 10 days of post-surgery testing. The solid dark line represents the combined sham animals. There is no difference between sham, TBI, or milnacipran, so we pulled them. The dotted line is TBI animals that received vehicle osmotic mini pump administration, and they, on multiple days post-testing, they perform significantly worse than sham by necessitating more trials to reach criterion to finish the 4 sets every day. The hashed line represents injured animals that received milnacipran. And you can see that on many of the test days, they were significantly better than vehicle TBI. Although it was not significantly better than sham, on a few of those days, they seemed to be even a little bit better than sham. Nevertheless, the milnacipran restored cognitive function in injured rats after frontal lobe injury. A similar pattern we see for total errors. So the vehicle TBI group, the dotted line, was significantly worse than sham and significantly worse than their own baseline too, for that matter. Whereas chronic milnacipran significantly improved performance compared to the vehicle-treated injured animals by rendering less number of errors. So after they finished the operant set shifting test, the same animals underwent training and testing for the digging set shifting test. And we see total trials on the left, total errors on the right. And we found that vehicle TBI animals performed significantly worse on the first reversal for both total trials and total errors, as well as on the third reversal, whereas injured animals that received milnacipran had significant improvements on the first reversal for total trials, and there was a trend on the first reversal for total errors as well. So we're also curious to see how these animals performed individually from between the two tasks. And again, this hasn't been done. So the way that we found the most suitable was to plot on the y-axis the average total of trials post-surgery for the operant set shifting task, and on the x-axis the total trials added, basically cumulative, for the digging set shifting task. And although it's not, it's more of a qualitative analysis, but we wanted to see how they cluster. So the black dots represent the combined sham group. The red squares represent vehicle-treated TBI animals, and a good percentage of those TBI animals are outside of the sham, suggesting that they tend to perform worse on one task, they perform worse on the other as well. And the purple triangles represent the injured animals that received milnacipran, and they cluster more closely to the sham group, again suggesting that when they had improved performance on one test, they also have improved performance on the other test. So moving on to the last main part of the talk, as I presented up until this point, we had normal cycling females and males. They're all young adults. We are also very interested in continuing the studies and looking at females as well, or we're completing the operant set shifting study with females. We're also currently looking at aged rats because, as I mentioned at the beginning of the talk, the elderly are a big risk group for traumatic brain injury. And we are lucky to have a governmental grant from the NIH that allows us to look at aged rats this year. But one of the groups that is susceptible to injury, as I mentioned at the beginning, is children. And more than 500,000 emergency department visits every year in the United States are from children sustaining traumatic brain injury ranging from mild to severe. So we wanted to look at control cortical impact, pediatric injury effects in an instrumental learning task, which is basically a task of goal-directed behavior, as well as digging set shifting task during adolescence. So we use parietal injury at postnatal day 17, which is the equivalent of a toddler basically in a human. And in the operant chamber, there is no major rule for this task. Basically, it's a fixed ratio of 1. You put the animals in for 12 days, and they learn that when the cue comes on in the center, it just stays lit. Whenever they poke their nose into it, they'll get a sucrose pellet. And as the days go on, they learn to do that more and more. They can go up to 100 trials or 100 pellets per day. After they finished the 12 days of instrumental learning, basically at postnatal day 42, which is in the middle of their adolescence period, they were habituated, trained, and testing on the digging set shifting task. So the instrumental learning task, again, looking at goal-directed behavior post-pediatric TBI, We have on the left panel total trials, and on the right panel task irrelevant pokes. I'll describe those in a second. So we actually hypothesized that injured rats that would have received the injury during the pediatric period, whenever they're tested for this task during adolescence, they would perhaps engage in less total trials than the sham rats. However, as you can see on the left graph, it was exactly the opposite. So the sham curve, which is in blue, is pretty normal for what you'd see in this task. As the days go on, the animals start maximizing towards 80, 90, close to 100 trials. However, the TBI animals performed significantly more total trials than sham. It was an overall group effect as well as on individual days 3, 7, and 8 for post hoc analyses. So that was interesting because they definitely do not appear to have a motivation problem. The computer also assesses how many times the animals are poking on the left and right cube portholes, although they really don't do anything. So they're not receiving a cue, there's no punishment, so there's really no outcome for those, but you still measure them as a task irrelevant poke, which is typically seen as a measure of exploratory drive. The typical adolescent animal would be curious to see what those pokes do. The blue curve on the right graph is the sham animals, and that is very normal according to the literature. As the days go on, considering that those portholes don't really have an outcome, it's almost like an extinction. So the normal sham animals would just start paying attention less and less to those portholes. However, our TBI animals from the very beginning paid significantly less attention to those portholes. Basically, although they poked more in the center, they paid less attention to the other holes, suggesting either possibly reduced exploratory drive, but also less distractibility. A lot of clinicians that I've discussed these findings with see it as a potential of kind of ADHD findings where children would spend a lot of their energy trying to focus on their necessary task at hand, therefore perhaps not having the ability to multitask as much. We also looked at premature center pokes, and there were no group differences. And we looked at poke to pellet latency. So once they poked instrumentally, how fast do they go to retrieve their pellet? Normal sham animals, it takes them about 4 to 5 seconds, which we see in the blue curve. And again, the TBI animals retrieve their pellets significantly faster than sham. So again, there's really no motivational deficits. There was an overall effect group as well as individual post hoc differences for days 3 and 11. They really, really wanted to go their pellet very fast. When we looked at the digging set shifting task, assessing executive function during adolescence, post-pediatric TBI, there are actually no group effects. But it's interesting because it doesn't mean that the effects are not there. Once you would go back to the slides from the adults, one would notice that normal sham or control adults actually have less total trials to criterion than our sham adolescents. Normal sham adults are typically in the low teens, whereas the sham adolescents are around 20-22 for trials to criterion, which could be a reason why we didn't find a TBI effect. But this higher baseline in adolescence is normal. It has been reported the first time by Dr. Jill McGahey at University of New Hampshire, basically that adolescent animals have more cognitive rigidity than adults. And they make more mistakes. Also possibly because they're so curious with their environment and they inhibit impulsive choices less than adults. So for this particular study, we are very interested in assessing behavior for the digging set shifting task in animals that were given the same type of pediatric injury, but allowed to develop to a young adulthood and see if we're able to tease out injury differences at that age. So to summarize, we provided here, and again, sorry for the beginning slides that were somehow timing themselves at a few seconds and moving forward. We provided the first studies to implement sustained attention, operant set shifting, instrumental learning testing in the traumatic brain injury field, as well as, to the best of our knowledge, the first study to use 2 executive function test modalities incorporating different salient stimuli in the same rats. As I mentioned, the operant chamber is great because it is an objective way for the animal behavior to be assessed, whereas the digging set shifting task allows for the natural sensory interest of the animal to take hold, though it is a bit more difficult because the experimenters have to be highly objective and highly trained to basically assess all the animals the same way. We found that frontal lobe and parietal lobe TBI induced modality-dependent deficits in both operant behavioral tasks, as well as a digging set shifting test. Different drugs such as galantamine augmenting acetylcholine, citalopram that augments serotonin, and milnacipran that augments serotonin and norepinephrine attenuated the functional deficits incurred after different injuries to the either frontal or parietal cortex. We found divergent alterations of goal-directed behavior in adolescents subjected to pediatric parietal TBI. And in ongoing and future studies, we will continue to elucidate possible mechanisms underlying such deficit across the lifespan, ranging from pediatric and adolescents to aged animals. For sake of time, there's no time to basically show some of the other findings in our laboratory that use the tissues and the brains from these rats, but we implement histological and molecular biology approaches to work towards these goals, such as Western blotting, immunohistochemistry, proteomics, and oxidative stress. So I thank everyone for your attention, and I would be happy to entertain any questions that might be sent my way. Yes, and lastly, this is my laboratory. I would like to state that starting next year, we have a postdoctoral position available. So if any proactive graduate students out there who are looking to graduate at the end of this calendar year or early next year are interested in our type of research, please feel free to email me. You can see here on the left side a wonderful group of our current students and technicians and residents in our laboratory. And of course, thank you to our former team members, to our collaborators, and to our funding from various sources, ranging from local at University of Pittsburgh to the National Institute of Health. Thank you again.
Awesome. Thank you so much for that fantastic presentation, Corina. It was really great to hear about your work, and I'm super excited to start this Q&A session. Okay, so let's kick off this Q&A with our very first question. The first question is, have you looked at estrus phase in females, and are there any behavioral outcome correlates?
Thanks, Sarah. That's a great question. As I mentioned at some point, we look at normal cycling females, particularly because in previous research from our center, as well as from others, motor and spatial learning activity was not different depending on estrus phases in females. However, in one of our digging shifting test studies, we did look at estrus phases. And again, injured animals performed worse on the digging AST, regardless of whether they were cycling in the estrus, proestrus, or diestrus at that time. Further suggesting the fact that the different estrus phases might not affect performance on the task, and they would all be sensitive similarly to injury. But in our subsequent studies using operant chambers, because it's the first time when we assess operant behavior in females after TBI, we definitely plan on looking at the estrus phases in that as well and reporting them accordingly.
Great. That seems like a really awesome avenue for future studies. We have a second question here. This question is, what is the frame of thought for both frontal and parietal brain injuries to lead to similar executive function and behavioral flexibility deficits?
Thank you, Sarah. Another good question. So, as I mentioned before, clinical findings suggest that patients with different types of injuries, so basically injuries to different locations of their brain, render impairments in a lot of these neuropsychological tests similarly, right? Whether it's the Wisconsin card sorted test or the NIH toolkit battery test, which allowed us to start investigating these tasks and looking at different types of locations for the injury. We indeed are not able currently to separate facets of behavior depending on where the injury was from. But I don't think that that would necessarily be the goal, because I guess one gross kind of way to consider it is, one can imagine that finger dexterity could be affected similarly if one's elbow or shoulder are injured, right? So considering the afferent innervations to the frontal lobe from different parts of the brain that also pertain to different neurotransmitters, right, ranging from acetylcholine, dopamine, serotonin, norepinephrine. Once you injure different locations in the brain that lead to the frontal lobe, or whether you injure the frontal lobe directly, you could have similar behaviors. What would be really interesting as we continue our studies is to look at how perhaps different treatments might lead to various levels of improvement depending on where the location of the injury is, as well as histological and molecular biology assessments of life cycles of neurotransmitters and determining how different brain regions might differentially express certain markers differently based on where the injury came from.
That makes a lot of sense and is a really good question. So thank you for that answer. We have another question here. This question comes from Ross. Ross is asking, do you have recommendations for attentional tasks that translate well to mice?
So a lot of these tasks have been successfully performed in mice as well. So, for example, the digging set shifting task was initially developed for rats. However, circa 2008, 2009, different groups, especially Jared Young, Mark Geyer, so kind of the University of San Diego cohort, behavioral neuroscience cohort, they successfully started publishing in mice as well as other groups. So currently, I would say that this task can be successfully implemented in mice as well. And there has been a variety of operant chamber, I guess, versions of attentional tasks, including the newer touchscreen tasks that you can find in the literature, in more recent literature, that they're successfully used in both rats and mice, which is great.
Cool. So that'll be really helpful for Ross. Hope that answered your question, Ross. We have another question here from Evelyn. I think I'm pronouncing that correctly. Why did you use the 3 CSRT instead of the 5 CSRT task?
That's a good question. So, one, because I was trained as a postdoc in Dr. Bida Moghadam's laboratory using the 3-choice serial reaction time task. So it was a little bit easier for me to start implementing that task. 2, as far as what we wanted to accomplish, aka TBI-induced deficits of performance, we felt, based on the literature of both the 3-choice task and the 5-choice task, that we would be able to achieve that regardless of which of the 2 tasks was used.
Okay, that makes sense. We have another question here from Damien. His question is, what is the grade of the injury in this experiment? Is it mild or severe?
Great question. So based on the first digging set shifting task graphs that I presented with the different types of injury, we found that the more moderate or moderate to severe injury were the ones that rendered reliable and replicable deficits in these tasks. So if you remember also the cortical lesion volumes at the end of that one study, again, it was the 2.6 and the 2.8 millimeter cortical deformation group for the 2.8 and the 3.0 cortical deformation group for parietal. 2.8 millimeter cortical deformation depth has been the standard in many laboratories before we started using these tasks in the TBI field. And for the frontal lobe, again, because you have a smaller brain region, we use the 2.4 millimeter cortical deformation depth that renders similar deficits of performance in the digging set shifting task. So overall, yes, I would say that they're categorized as more moderate or moderate to severe injury. At the same time, though, without rendering death in these animals.
Gotcha. Okay. We have a question here from Scott. This question is, based on the anatomy that you presented initially involving frontal subareas, how was damage to the parietal cortex producing specific impairments in set shifting?
The way that I always saw it was kind of like a big train hub. So when you block the train hub with the trains that are going towards the frontal lobe, you basically affect multiple neurotransmitters that lead to that brain region. So because of the level of the cavitation that the moderate to severe injury renders whenever you give parietal injury, it is to be expected, and we are currently assessing that with the variety of histological techniques that I mentioned earlier. We're basically assessing the level of disruption of a multitude of neurotransmitters in the frontal lobe that might have been affected. When you basically render such a big injury to the parietal region, one can expect again that it's not going to be just serotonin or just norepinephrine or just dopamine that will be affected, but rather all of them to a certain extent. And as I mentioned, in the multitude of studies of digging AST that were done prior to the test being involved in TBI, all of these neurotransmitters have been previously determined to play at least a partial role in these tasks. So I would say that at this point, just based on the lesion study, it's difficult to say exactly which type of neurotransmitter may have affected, but based on our therapeutics and current histological assessments and molecular assessments, we are looking forward to be able to tease out again in more details the level of different neurotransmitters that were disrupted in the frontal lobe.
That's amazing. That's going to be really cool. Thank you for that answer. Before we continue with our Q&A, we still do have a couple minutes left, so we'll answer a couple more questions, but I did want to bring your attention to the survey that appeared on your screen in the last minute or so. If you do have to leave a little bit early, please fill out that survey before you go, and if you can stay for the Q&A and the rest of the Q&A, that'd be much appreciated. So, Corina, we're going to continue with our next question now. This question is from Hugh. He's asking, does the CCI-related anxiety affect the performance of rats in AST?
That's a very good question, and I didn't get a chance to show some of our tests of effect because we're also doing those, particularly because I wanted to focus this talk more on executive function and behavioral flexibility tasks. But yes, so not only do humans present comorbidity of depression and anxiety and cognitive deficits, but the cognitive impairments may and are known to be augmented for sure by alterations in mood and affect, which basically means higher anxiety. And in some of our studies, we are finding that injured animals do present with higher levels of anxiety as determined on tests such as the open field test, elevated plasmase test, as well as the shock probe defensive bearing test, which basically allows the animals to engage in either passive coping, sitting immobile in the corner of the cage, or active coping by pushing bedding over this electrified probe that had just shocked them. So you can basically see a shift, a stress-related shift from active coping to passive coping and vice versa. And we're seeing injury negatively impacting affect as well. So based on the clinic, it is to be hypothesized that the level of anxiety may affect their decision-making process. So although our animals are not too anxious to the point where they would sit in a corner and not perform at all, because in that case, you can't really assess performance when you do not have trials at all. But it could have affected their decision-making process, therefore contributing to impaired performance.
Okay. We do have a couple more questions, so I'm not sure if we're going to be able to get to them all, but I'll keep pushing through. So this question is from Damien. He's asking, the effect of milnacipran is very interesting. Is it already prescribed in TBI patients, including young patients?
There have been a couple of clinical trials in which some of the outcomes seemed promising. But as I mentioned, it's kind of more anecdotal in terms of the fact that it is FDA approved for fibromyalgia, but it is not one of the first lines of treatment that doctors would use for either just, depression or cognitive deficits with an unknown etiology, or whether it is known that depression or cognitive deficits have risen from a recent traumatic brain injury. So to answer that question, a couple of clinical studies, they were not young patients, but it would be very interesting to see that in the near future.
Okay, and I think this is going to be our last question, just to be respectful of everyone's time. This question is from David. He's asking, do you have any information on the effects of group housing versus single housing on TBI testing?
So environmental enrichment housing has been shown before by a variety of groups and a lot in our center by my colleague and former mentor, Anthony Klein, that it is highly beneficial for alleviating particularly spatial and learning and motor deficits after TBI. We started in some of our studies to implement environmental enrichment either alone or in combination with other treatments, as I shown in the study with citalopram, to determine how it may be efficacious in reducing cognitive deficits on the higher order executive function tasks. Now, we did have a study before from the Klein laboratory where they basically separated the different components of the environment enrichment to determine whether it's group housing or whether it's the toys that are stimulating the animals and so forth, or the size of the cage with the different levels. And they found that while each of those components played a partial role in reversing TBI-related deficits, it was really the combination of all the factors that rendered the most pronounced beneficial effects. So yes, group housing does play a role, but it's not the only factor to get the entire kind of rehab-like component of enrichment. You do need the sizing of the cage and the toys that they are changed daily to stimulate the animals, besides the group factor.
Okay.
That was a great question.
Yeah, really awesome. Thank you so much for your wonderful insights, Corina, today in the Q&A, as well as your presentation.